Alright, so this video is all about Punnett squares. And the big idea here is I want to introduce a lot of vocabulary that's going to be important, um, and then get into some examples of how they actually work. So with Punnett squares, the idea behind them is to determine the probability of traits in offspring. When we talk about classic Mendelian genetics, we're talking about inheritance. So we still can't forget that all of this comes back to genes, and genes are sequences of DNA that determine traits. So the DNA helps make RNA, the RNA makes protein, the protein determines your traits, like whether you have brown eyes or, red hair or whatever. So our first important terminology is genotype and phenotype. So when we're talking genotype, we're talking about those genes. And the phenotype is like what you actually see, the physical characteristic or the trait that comes about. Now, when we talk about alleles, we might be talking about a particular trait, like flower color. But alleles are different versions of that flower color. For example, in this example, it's violet and it's white. So flower color is the trait, and the alleles are violet and white, and what it actually looks like is its phenotype. But if we go back to the genotype, we abbreviate the gene and what's in the genes with letters. And those letters aren't the same as like when we're talking ATGC. We use other letters for this. And in this one, I'm going to talk about A's. So if we look here, and we're thinking about what Mendel actually did. He was a pea breeder, and he had this experimental farm, and he crossed all these purples, purple into purple into purple into purple and purple. They always came up purple. And then he also had white and white and white and white and white and white, and they always came out white. So if something is homozygous, we'll say it's a purebred or it's pure breeding. When they're crossed, they always get the same phenotype as the original phenotype. And we abbreviate that homozygous, the genotype, as like big A, or little a. So both alleles in both chromosomes for that trait are the same allele. So it's big A, or little a. When we are talking heterozygous, it means that different alleles are present in the genotype. So we're not talking phenotype yet, but in the genotype, we're abbreviating that the gene for color as with an A. That's the letter we're choosing to use to represent the genotype for flower color. Now that could be a big A or a little a, and that means that two alleles are mixed in that organism. When we talk about dominance, when something is dominant, it's which allele is present in the phenotype of a heterozygous genotype. So if you have something that's mixed here, so these are purebred, but you mix them together and you get heterozygous genotypes in the offspring. And whichever one shows up in that first heterozygous generation, we say that whatever one shows up would be the dominant trait. So if we were to assign letters to these, this would be big A for the purple and little a for the white. And again, defining these terms, the recessive allele here is the one that's carried or hidden in the heterozygous genotype. So these pea plants are still carrying alleles for white flowers, you just don't see them. So they can pass on this white allele, and we consider that a little a. So the one that doesn't show up when it's heterozygous is the recessive version of that trait. And the only time you can see a recessive in the phenotype is if it is homozygous in the genotype. So again, we're just got to use the words. So this white flower has to be little a, otherwise you wouldn't see white color. It would turn up purple. Okay, so let's say we're looking at this, and now we're tying this into the world of Punnett squares. So in this, we've got a white flower, little p, crossed with purple, which is big P, big P. So the phenotype of this is white. The phenotype of this is purple. They are both homozygous, and in this case, it's homozygous dominant because it's capital letters, and this is homozygous recessive because it's little letters. All of these possible offspring are heterozygous because it's a mix, it's different between big P and little p. Big P, little p, big P, little p, big P, little p. So that's where we get into this world of heterozygous. Now, what is dominant? The purple is dominant because when it is heterozygous, big and little, the purple is what shows up. And the recessive would be white because when it's heterozygous, you do not see the white color in the flowers. Now, in this example, the genotype for this is big B, little b. The genotype is the letters that represent the genes or the code. This one is big B, little b. So those are the genotypes. The phenotype of these parents are both purple flowers. These parent flowers are both heterozygous because it has a big letter and a little letter. Even though the parents are heterozygous, they can produce homozygous offspring. So when they cross this chromosome with this chromosome, you combine them together to get big B, and that is purple. Again, these heterozygous are carrying or hiding the allele for the recessive trait, but if we match up the chromosomes that are for the allele that's recessive, these can come together and you can get a homozygous recessive offspring, which would then show up with a white phenotype. They can also cross this big B with this little b, and you get a heterozygous offspring, which would demonstrate the dominant phenotype of purple. And it's the same idea here. Big B, little b, and you get heterozygous offspring. Okay, so now to expand on this a little bit and to just try and, got to make it a little bit harder, we look at all of these chromosomes. And if we are talking about classic Mendelian Punnett squares, the examples I want to show you are involved with dihybrid crosses. Instead of just looking at flower color, we can also look at different other traits as well. Now, Mendel got incredibly lucky. He looked at 7 traits. They all existed on different chromosomes, which makes things so much easier. Now, you can see this Punnett square. This box is bigger. And the one thing I didn't talk about with the other Punnett squares, those other boxes, is the whole idea is you're trying to find the probability of the offspring. So again, we're going to look at this dihybrid cross. So it's 2 traits, and it's in the P1 cross, this first parental cross, and it's purebreds, true breedings crossed with each other. We're going to look at 2 traits, flower color, but now we're going to look at green and purple, and plant height, dwarf and tall. And we're going to use G's to represent flower color and D's to represent plant height. The big G is going to be the dominant green, and the little g is going to be the recessive purple. the plant height, dwarf is dominant, and tall is recessive. So we have one parent. The genotype is big G, big G, big D, big D. It's diploid, which means that even though we have one gene, we have a backup copy. That's what diploid means. We have two chromosomes that carry the G alleles. And one is big G and the other is big G. They're non-linked, and we won't quite get into that, but that means that they're on separate chromosomes. And yeah, that's another day. But big D, big D. So this is homozygous green, homozygous dwarf. The other parent here is going to be little g, little g, little d, little d. So again, they're non-linked, which means they exist on different sets of homologous chromosomes. One is going to be little g, and the fact that it's diploid means that we don't just have one set of each chromosome, we have the backup copies. Now, what goes on the outside here of the Punnett squares are all of the possible gametes that can be created from these chromosomes of the parents. And when we say this is diploid, we're saying that there is backup copies for all of these original genes. But when we have gametes, they don't have backup copies. And gametes we typically think of as sex cells. So in the case of peas, it's pollen, not sperm, but it's the same idea. So you could have one G from these two, and you pair it with one of the Ds from this chromosome. And that contains all of the genes, but only one copy of each. So you have to have a G and you have to have a D. G and D, G and D, G and D. So a lot of times when I teach this, I say, let's name the chromosomes. You have 1 and 2, and then you have 3 and 4. So to make sure you got all the combinations, sometimes it gets a little bit tricky. We go 1 and 3, 1 and 4, 2 and 3, 2 and 4. So you'd pair up the first 1 and 3, then I'd pair up the outsides 1 and 4, then the insides 2 and 3, and then the outsides, the far to the right, would be 2 and 4. And you do the same thing on the other side. G and D, G and D, G and D, G and D, G and D. When you cross these, then we get the next generation. So these are all possible gametes. The inside of the box is all of the possible zygotes or offsprings or fertilized egg and sperm combinations. We call that the next generation. From P1 is F1. So you take the big G and you match it with the little g. And you take the big D and you match it with the little d. And therefore, the genotype of one of these offsprings is big G, big D, little d. The reason I have all of these things in here is to just get you thinking that it's not just letters. Those letters represent genes, and those genes exist on chromosomes. So again, we started diploid with backup copies here, right? And then we had haploid, half the number of chromosomes in the gametes. But when fertilization happens, when egg and sperm come together, we're back to diploid, back to backup copies. So if you start with 4 letters, the inside of the boxes have to be all be 4 letters. And G goes with G, and D goes with D. We're matching up this column with this row. Those letters go in the box. And we do the same thing again and again. And because all of these gametes are the same, it ultimately comes out that they're all heterozygous, because we start with homozygous dominant on one side, homozygous recessive. You combine all of that, you get all of them being heterozygous. which are a combination of big and little genotypes, dominant and recessive. So then what are the phenotypes of this whole generation? Well, big G rules over little g, so they're all going to be green. And big D is dominant over little d recessive, so every single one is going to be dwarf. So all of them are going to be green dwarf. Now, let's say we take these F1s and we cross them up to get an F2 generation. So both parents now are heterozygous, big and little, big and little, big and little, big and little. And now we want to get to F2, the next generation on. So again, we have our big Punnett square. This is the F1 cross. We're crossing big G, little g, big D, little d. Same thing with the other side here, big G, little g, big D, little d. Again, we're making gametes, which are half the number of chromosomes, and that happens through meiosis. We've done a lot of mitosis, but now this is meiosis. So we go 1 and 3. Remember how I said big G, big D, 1 and 3. I go 1 and 4, 1 and 4. I go 2 and 3, and I go 2 and 4. And then I move on over here, same thing. I need a G, I need a D. 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 So I'm going to always on these put the G's first and then the D's regardless of the capital or lowercase letters. When we go here, I'm going to put the G's together and the D's together. And if they are heterozygous, I'm always going to put the capital letter first. But the outside, again, remember, what is the outside of the Punnett square? It is the gametes. What are all of the boxes on the inside? Possible zygotes. What's our F2? We bring down a G, we bring over a G. Bring down a D, we bring over a D. So the first genotype of that first box is big G, big G, big D, big D. And we keep going. Bring the G's, bring the D's. Bring the G's, bring the D's. And again, even though I usually bring the top down and then the side over, this big G, I always put it first. So I keep going. Put the G's together, put the D's together. I just keep going through and filling in every box, matching up what goes, which columns go with which rows, and putting those letters in the boxes. And now, I said, the whole idea behind this is to try and determine the probability of the offspring. So what are the F2 phenotypes? What are all the different physical characteristics that could show up in these offspring? Again, let's remember, big G is green, little g is purple. Big D is dwarf, little d is tall. So we could have green dwarf, green tall, purple dwarf, and purple tall. These are all the different combinations we could have. So which is which here? If it's going to be green, it has to have at least one big G. If it's going to be dwarf, it has to have at least one big D. If it's going to be tall, it has to be little d, little d. If it's going to be purple, it has to be little g, little g. So it ends up like this. All of these green dwarfs, there is at least one big G in all of these, and at least one big D in all of these. Green tall, it has to be green, it has to have a big G, at least one or two. In order to be tall, it has to be little d, little d. That's the only way the homozygous, the only way the recessive trait shows up in the phenotype is if it is homozygous. Again, lots of new words here. Just try to use them as often as possible. Here, these ones are purple because it's little g, and they're dwarf because they have a big D, at least one big D. To get purple tall, it has to be little g, little g, little d, little d. So this would be a 9 to 3 to 3 to 1 ratio of offspring for the different phenotypes. And that's eventually where we need to get with your ability with making Punnett squares.